The scientific method often begins with an observation that raises a question. Next, a possible explanation called a hypothesis is formulated to answer the question. But to be useful, that explanation must be testable. Generally, the hypothesis will lead to a prediction. The prediction is tested by experimentation or further observations. The results are then interpreted to form a conclusion. If the results support the hypothesis, new predictions can be made to further test the hypothesis. If the original hypothesis is not supported, an alternative hypothesis is generated and tested. Here's one example of the scientific method in action. If, while walking through the rainforest, you came across plants which you later identify as lemon ant plants growing alone with no other plants growing around them, you might record and describe that observation. Comparing the density of different plants in the patch to the density of different plants in most other areas of the forest floor, the relative isolation of lemon ant plants strikes you as odd. You collect information such as the area of the patch, the approximate number of plants in it, and the size of the cleared area around each plant. You also look for another patch, make the same measurements, and compare the patches to see whether there's some correlation between the size of the patch and the area of the clearing. Your measurements suggest that the larger the patch, the larger the cleared area. Next, you develop a hypothesis as a possible answer to your question, asking why lemon ant plants are found in otherwise cleared areas. Your first guess is that the lemon ant plants are producing a chemical that inhibits the growth of other plants. To be useful, this hypothesis needs to be testable. Next, you make a prediction based on your hypothesis and test it with a controlled experiment. You might predict, for example, that if your hypothesis is correct, that a chemical extract alone, in the absence of the intact plant, will inhibit the growth of other plants. You therefore select one lemon ant plant and liquefy its tissues in alcohol using a blender to produce an extract of all the chemicals in the plant that are soluble in alcohol. To discover whether those chemicals inhibit the growth of other plants, you prepare two identical plots of ground and seed them both with the same variety of plants. You then spread the plant extract over the experimental plot, but you leave the other plot, called the control plot, untreated. Sure enough, no new plants grow in the area that you treated with the alcohol extract, but plants grow well in the untreated areas. Do these results support your hypothesis? They seem to, but it's possible that the alcohol in the extract is preventing the seeds in the experimental plot from growing. In this experiment, you actually had two variables that were being tested, the alcohol and the extract itself. You need to experiment again, this time with two control groups and one experimental group. In each plot, you add seeds, but you treat one plot with the alcohol extract, you treat another plot with alcohol only, and you leave the third plot untreated. Sure enough, plant growth is retarded only in the area treated with the extract. Plants grow in the plot treated only with alcohol and in the untreated plot. Your results indicate that the chemicals in the plant tissue are inhibiting the growth of other plants. This, in fact, may be your conclusion, that the lemon ant plant produces a chemical that inhibits the growth of other plants. Note, however, that although your results support your hypothesis and by extension your conclusion, they do not prove the hypothesis. For example, perhaps your results could be explained in another way. Perhaps the lemon ants that live on the plant deposit a chemical on its surface that washes off with rain and inhibits plant growth in the surrounding area. The ant-produced chemical might also have gotten into your extract, along with the chemicals produced by the plant itself. A hypothesis can be proven wrong when the results do not support it, but results that are consistent with a hypothesis do not prove that it's correct. Even successful experiments often lead to revised hypotheses and further experimentation. What if the plant extract had not inhibited the growth of other plants? This would mean that your hypothesis needs to be rethought and that you may have to find another explanation. Essentially, you must now look for new variables.